target discovery and optimization is one of the major components used for drug discovery pipeline. In target discovery, we have two broader categories, which are physiological based approaches and disease centric approaches. Now if I want to elaborate these approaches, then we have four further categories, which are basically structure based approaches, cell based approaches, genetics based approaches and target class based approaches. अब ये approaches हमारे पास क्यों आई and why we have a good way to explore further details. One major contribution was Human Genome Project, which gave us very huge amount of information to play with different genetic and proteins information. Then we have high throughput experiments, where we can perform our experiments at a very big and large amount of data instead of performing experiment with single genes or proteins. So I will start from structure based approaches, that what are those structure based approaches and what is the usefulness of these structure based approaches. Structure based approaches are majorly used for protein sequences, although they are used for gene information also. And what is the basic concept behind these approaches is that they work on the contours of the cognate surfaces of lesions. So what are contours and what are cognate surfaces? Contours are basically outer part of the lesions, and cognate surfaces are those surfaces which are similar across different lesions. अगर हम ये find out कर लें कि what are common surfaces or what is the common structure between two lesions, then we can find out similarity and dissimilarity between these lesions. And this information is used while developing those drugs which are based on structure based approaches. So what happens is, what is the advantage of this structure based approach is that we have a good record of potency of the medicine plus selectiveness of that medicine. So potency is basically power of medicine, and selectiveness means that in which cases this drug can be used. So based on this definition, there are some common drugs which are available in market related to this structure based approaches. I have given some names here. Out of these four drugs, I will say that captopril, which is written second in this bullet points, it is used for curing of hypertension. Similarly, this zanamivir, which is written in the fourth place, it is used for our influenza viruses. So these structure based approaches and drugs based on these structure based approaches are common in the market. Now we come to protein structure based approaches. As we said that there are ligand based approaches which are based on genetic information. Then we have protein structure based approaches. Drugs made based on these approaches, there are again 5 different categories. Number 1 is where we have target identification from sequence structure and homologue recognition. Homologue, where structure is quite similar. Then we have those information which is based on structure genomics and drug effects. So structure genomics which is not probably homologues, which may have different genetic structure. Then we have identification of ligand binding region. So ligand binding, as ligands are binded on chromosome and they have their specific locations. So ligand binding can be at different places, and when this ligand binding is at different place, the working of that medicine becomes different. Then we have identification of hits and leads, that is basically concept of combinatorial chemistry, where we have combination of compounds and which have if they are similar they are put together, if they are different then they are separated apart. Then we have structure guided design and screening, which is basically used in NMR techniques, which are basically nuclear magnetic resource resonance techniques, and these techniques are used there. Then we have kinase drug discovery and kinome. So kinome and kinase, as you know, kinase are basically our enzymes which are used for different purposes, and kinomes are discovered kinases which are roughly at the moment 50,000. So these are used and they are coming under the heading of structure based approaches. Then we have cell based genetics. Now in cell based genetics, we want to understand the complete structure and components of cells. Now what would be the effect of a complete cell information on phenotypes, and what would be the effects of individual components of a cell on phenotypic traits? That is basically purpose of cell based genetic approaches. So there are three common divisions of this cell-based approaches. Number one is expression profiling. This expression profiling, it is basically a proteomic approach where proteins information is used to identify disease-related genes, and these related genes, how they are discovered using differential expression profiling, homology, and post-translational modification. So what we have cells, and then we want to identify what are different effects on different phenotypes. Then we have biochemical and cell biological assays. So these cell biological assays, basically instead of working on single cells, we have assays which are consisting of multiple cells, and then we want to find out what are the effects of those cell assays on drug targets discoveries. Then we have cell-based genetics. So this cell-based genetic discovery, what happens in this one, this works by changing different things inside the cell. For example, it will work out by correlation with the phenotypes, and then it can disturb or change the genetic behavior of a cell, and then it will see how the drug is working on different phenotypes. Then we have cell-based genetics. As I said, that it works by working or changing different mechanisms inside the cell, such as transmission, transformation, activation, migration, or similarly host of biological processes which are related to some human organism. So that was our cell-based technique, that is a second technique. Then we have genetic-based target identification. In this genetic-based target identification, what we want to identify that what is the behavior of genes while target discovery. There are two major concepts which are part of this technique. Number one is position cloning. Position cloning, basically it is a laboratory technique which is used to find out the position of disease associated genes on a chromosome. So what are those genes which are responsible for a disease, and we want to find out the position of those genes. The other thing is candidate gene approach. In candidate gene approach, we want to find out our SNPs so that we can, with the help of those SNP, we can find out which genes are being responsible for for disease generation. So in human genome project, there were roughly 10 million genes. Out of those, 3 million have been identified for this purpose. Moving on, we have finally target class centric approach. This target class centric approach, it is basically based on mechanistic approaches such as our ion channels, receptors, and GPCRs, which will be seen in some other modules. And then we have in gene production, we have roughly 24,000 protein coding genes. On the other hand, we have only 2,400 out of those 24,000 genes. 2,400 are or can be used for target discovery only. So how to describe the best candidate? So best candidates are defined or selected based on any of these different categories which can be used for target discovery out of these 24,000 proteins.